All right, so it looks like we have a critical mass. Welcome everyone to our first cardiology grand rounds of the year. Welcome especially to our fellowship candidates who are interviewing. We're delighted to have you with us, albeit virtually. This year, we're delighted to have Dr. Robert Gerszten, our Chief of Cardiovascular Medicine, give our cardiology grand rounds, and Dr. Jeremy Robbins from our division will be introducing him.
Thank you, Arti, and I second the welcome to all the applicants tuning in and to everyone else. It's really my great honor and even greater pleasure to introduce Dr. Gerszten for this, to kick off this year's cardiology grand rounds. Dr. Gerszten's education and training have brought him across the country to prestigious institutions like UCSF and MGH. But everyone knows that Rob's heart lies in Dixieland, where he's a native Virginian and proud alum of UVA. I think that background has contributed to Rob's deep sense of history. And so it's only fitting to watch Rob absorb the historical moment of UVA becoming the first #1 seed in NCAA history to fall to the mighty UMBC Retrievers in 2018, pretty soon after he came to the BIDMC. Thankfully, for all of us who work with them on a daily basis, this was quickly followed by their redemption national championship the following year. And I think that Dr. Gerszten's wide-ranging background and training suit someone whose academic contributions and achievements are simply expansive, running the gamut from making basic science discoveries in immunology and vascular biology, all the way to having the expertise, ingenuity, and really creativity to harness cutting-edge technologies in biochemical profiling and then bring them to the clinical arena in order to study cardiometabolic health and disease. But perhaps what makes Rob stand out above and beyond his many titles and honors is his infectious enthusiasm for science and discovery, coupled with his incredible commitment to his personal relationships. And this is best reflected by the decades-long, list of mentees that he has helped cultivate and watched flourish, culminating in his William Silen Lifetime Achievement and Mentoring Award, the only award I'll name here. And so I think that today's grand rounds topic, exercise as medicine, is fitting for someone who's built a rich career integrating chemical biology, cardiovascular physiology, metabolism, and has brought many, many mentees, including myself, along for the good ride. So with that, we look forward to your talk, Rob.
All righty. Well, thank you guys for that very warm introduction. I guess the timing for a discussion of exercise physiology is good because the Boston Marathon is going to be next month this year. So 4 years ago, I included some of our work on exercise and our kind of thoughts of how we were moving forward as we're about to embark on 2 pretty large efforts in human exercise in adults and actually in children as well. And I thought it'd be a fun time to update you on what hopefully you'll agree is some of our progress in the area. So I'm going to talk a little bit about, give an introduction, discuss some of the gaps in our knowledge, how integrative genomics might help fill in these areas, start off with some of our older work that happened to start in vitro in cells and animals, and then really move quickly to update you on work in 2 large exercise efforts. One I'll say is medium large, the Heritage effort, but it is the largest effort to date in the world. And then what's going to be the very largest study, almost a $200 million effort that's ongoing. And it's part of the NIH called the Motorpack, the Molecular Transducers of Physical Activity study, for which we are one of the biochemical profiling cores. And then finally, a little bit about future directions, how we're trying to synthesize our data. So observations relating exercise and human health go back a long, long time. Hippocrates said that eating alone will not keep a man well. He must also take exercise. William Heberden he of, I guess, nodular fame, also noted that sawing wood for half an hour a day cured a man of angina, maybe in one of the first N=1 studies. Jeremy Robbins reminded me of an observation that was made in 1953 by Jeremy Morris, who noted that bus conductors, the men, and we'll get to that in a little bit, who took all the tickets and ran up and down the steps of the double deckers in London, had lower rates of CHD than the sedentary drivers. Now I thought, maybe I should dig into that observations, because it seems so, who is this guy Morris? It seems so fraught with potential confounders. So I did a little bit of reading, and I really became a big fan of Dr. Morris and his work. So it turns out that Jeremy Morris was not his original name. As it turns out, he fled the pogroms of Eastern Europe in the early 1900s. His family did, and he ended up on a boat headed for Scotland. As they were arriving in Scotland, it turned out that the captain of the boat, well, his name was Morris, and the family thought that maybe they'd fit in easier if they adopted that surname. So that's where his surname came from. And then I kind of dug into the investigation a little bit and said, gosh, this is kind of it's a nice, cute observation, but how much meat is there really on the bones? And it turns out there was a formidable intellect behind the bones. So Jeremy Morris really thought a lot about confounders in studies. So he contacted the London Transit Authority that issued the pants that all of the workers in the transit authority used. So he tried to adjust for pant size, which is a reasonable approximation of waist to hip ratio. And then he was very intrigued as well by the conductors versus the bus drivers, and looked into their salaries. And eventually Dr. Morris became one of the early leaders in the field of looking at disparities among financial status and social determinants of health. So there's actually a lot more meat on the bones of this story. And it was really a lot of fun, and I really dove into it, and it's very interesting individual. Now, of course, more recently, there have been lots of more physiologically relevant relationships, associations that have been described between exercise, cardiorespiratory fitness, and mortality, for example. This is a classic study now 20 years ago from Myers and colleagues, in which they examined the relative risk of death as seen on the y-axis and quintiles of exercise capacity, both in normal subjects as seen in black, but in subjects, the white bars are subjects with cardiovascular disease, and you see across both of these groups a monotonic decrease in the relative risk of death. Importantly, this is this is a distinct study both for men and for women, although as we'll dive into later, this is clearly one area that's completely understudied in my opinion. I think the opinion of many others as well. So those are observations. Let's move on to interventions. This is a classic study as well from Bill Knoller, who's a collaborator of ours. We've been working with the diabetes prevention study. This is the study from which this figure comes from their first New England Journal paper with Dave Nathan and Bill Knoller and others, in which this high-risk group of individuals, it was quite heterogeneous group, more than 50% women, more than 50% of the individuals were non-white. There was a rather marked reduction in the lifestyle intervention. So in this case, it was both exercise and a more complete lifestyle nutritional intervention, but still a more striking effect than what was seen with the contemporary drug at that time, metformin, a 41% decrease in the cumulative incidence of diabetes in that study of about 3,000 or 4,000 individuals. Artie, do we have the interventional cardiologist? Is everyone on mute so they can't respond to this one? No, that's a joke. This is a small interventional study that I picked PTCA study versus exercise, lots of issues here too, but an interesting observation made in Circulation in 2004 in individuals, high-risk individuals. In fact, some underwent stenting, some underwent just exercise training, and in this study, event-free survival, again, many potential confounders, but was improved in the individuals who underwent in the exercise study. This is one of my latest, greatest, favorite studies. This is actually from another collaborator of ours, Ben Levine from UT Southwestern. This is a study of high intensity training that was really in typical Ben Levine style, is incredibly well done, incredibly meticulously phenotyped individuals, about 60 of them, who underwent a 2-year intervention of high intensity training. These were sedentary middle-aged adults. And you can see in the top panel in non-invasive assessments, and in the lower panel in invasive assessments, cardiopulmonary exercise testing with pulmonary catheters and wedge tracings as well. You know, you could see multiple metrics of lusitropy, of diastolic dysfunction, improved in this very high intensity intervention, suggesting that there could be a lot of possibilities here for individuals with heart failure with preserved ejection fractions. Now, one of the vexing issues in exercise physiology is that there's such a heterogeneity in exercise response. This is a classic paper from our collaborators Claude Bouchard and colleagues. This is from the Heritage study that we're going to dive into later. But for now, this is a 20-week intervention. And on the top panel, you can see from, I don't know how well you can see it, but it's a waterfall display of the almost, at that time, 600 participants and their change in VO2 max after the versus before the training protocol. In red are individuals who had low responses. Black was deemed average. Green was the high responders. And you can see really striking heterogeneity. Let's look at visceral adiposity, which is in the lower left. Overall improves. This was a rigorously done study in which weight was controlled, but adiposity, there was a heterogeneous response. On the bottom right is HDL improvements, more improvements than those who got worse. But again, if you look at any individual factor, any cardiometabolic trait, there's really a heterogeneity in the exercise response. Similarly, predicting who's going to have an exercise response is also a very difficult situation. Here we're looking on the x-axis at baseline VO2 max, and on the y-axis at delta VO2 max. And you can see that there's really no relationship here at all. And you can pick any trait that you like, blood sugar, insulin levels, et cetera, and trying to pick a response, it's really, really difficult to do. So that's really an important area, for which there's a gap in our knowledge. So what's the right intervention for the right goal? Does one size fits all? This is, from the American Heart Association, various websites, and, we're looking at all these 30 minutes of moderate exercise, maybe 5 minutes a day. Are we talking about the high intensity training that, I showed you in Ben Levine's intervention, et cetera, et cetera? So to kind of personalize this, I thought that, is this really something for me? What should I be doing? Should I be doing that 30 minutes a day at what used to be at the gym on the Peloton in my house and just watching CNN? Is that all that I need? Or as the third year fellows mentioned to me a while back during COVID when we met, they're like, Rob, I don't know, I don't know if they were thinking about my cardiovascular health or whether it was some sort of death wish, but they introduced me Tunde, who's one of the instructors on Peloton. We have a Peloton now in the basement. And I have to say, it became a near-death experience. But I will say, what's the right intervention? I think whether we need to work out with her or whether we can be more leisure, what's the health goal? There's a lot that needs to be done in terms of figuring all of this out. From a basic science standpoint, how does exercise impact changes on the nervous system? There was a paper in Nature Metabolism by Christian Wren just last week, in coverage in the New York Times, looking at proteins that may modulate the nervous system. There's effects on the immune system, bone, and a variety of cardiometabolic organs. And then how does that in turn confer the health benefits of exercise? So we know some of the arrows, but we certainly have a lot more to learn for sure. So if you are able to capture as many proteins and metabolites that are downstream of our genes and the transcripts, well, we would argue that that's a particularly robust or important way to begin to dissect the pathways that might be contributing to cardiovascular or cardiometabolic phenotypes, and to help us develop novel sorts of predictors. So genes make transcripts, transcripts make proteins, but the proteins and metabolites are really changing rapidly in the blood. And I'm not going to get into technologies today, but we're now assaying 7,000 proteins in the blood and up to 10,000 small molecule compounds in the blood as well. So instead of getting your Chem 20 on a patient, or maybe a slightly more detailed phenotyping, we're really able to capture thousands of analytes and really dive into biological pathways in ways that we couldn't have anticipated even 5 years ago, much less 10 years ago. Okay, so one of the areas in particular that's amenable to research, if you can look in the blood at factors that are changing relatively rapidly, is an area that's growing known as the exerkine research area, or things that are secreted during exercise, either acutely or chronically, that might confer signals to other organs and be part of a network, as we see on the right, of interorgan communication. And an area of particular interest in our group has been myokines. Things, again, these are sort of buzzwords and cute names, but any factor that might be generated, for example, by the muscle, that in turn might leach into the blood and confer effects. In this case, I'm going to show you, start off with some effects on adipose tissue biology that began in basic science work, and then we'll talk about human studies and how we try to integrate that. Okay, so why fat? Well, this is a slide that was given to me by my close friend and colleague, Paul Cohen, with whom we just secured an International Leduc Foundation grant to study adipose biology. And Paul always asks the question, is fat the problem, or could it be part of the solution? So broadly speaking, there are two types of fat. There's large fat droplets that are stored in white fat. White fat has few mitochondria and stores energy, really. Brown fat, by contrast, or beige fat, which is white fat that can be induced to take on some of the characteristics of brown fat, have numerous fat droplets, many mitochondria, and dissipate energy as heat via a specific transducer known as UCP1, or uncoupling protein 1. Okay, now the conventional notion is that this is really less relevant to adult humans, right? I mean, we've heard about brown fat, and undoubtedly many of you had, but it really, in children, hey, it's very active in mice. We are no longer babies. We're certainly not mice. Is this important in humans? And this is one of an example of several slides that came out about a decade, or papers that came out about a decade ago, including one observation by Aaron Sipos, who's actually a fellow at BIDMC, who showed that adult humans have cold-inducible brown fat. Okay, so actually many adults can have brown fat, and many have it at baseline actually, and it's particularly inducible if you stick individuals in the cold, but there are other inducers in adult humans as well. There's one study that from Paul that came out last year in Nature Medicine that went on to look at individuals from a number of data sets in which there was PET imaging. And Paul, who's an exquisitely fantastic basic scientist and clinician, really did as much adjusting as possible. A lot of these studies came from Cornell and from Memorial Sloan Kettering, where there was a lot of PET imaging done with as much adjusting as could be possible. It was pretty clear, even in the many normal individuals or seemingly normal individuals, there was protection from cardiometabolic disease in individuals who had cold-inducible, who had brown fat in those individuals. I'll end the talk by talking about the leveraging human genetics to get around this or to help us with this as well. But just I just wanted to kind of little bit of an aside of how exercise might be conferring some of its benefits and how we started off in the field, because one of the main regulators of fat metabolism and metabolism throughout the body and inducible by exercise is this transcriptional co-activator named PGC1 Alpha, which was initially described by Bruce Spiegelman, who is a collaborator of ours and is actually, a well-known National Academy member who's one of the fathers of energy metabolism, who identified this factor that is downstream of physical activity and then causes mitochondrial biogenesis, angiogenesis, modulates fatty acid transport, oxidation, etc. So what we did is, working with him, we began profiling just in a cell system. Could we see any differences in things that were secreted from muscle, getting at the notion of, can we find anything from muscle that might be modulating fat, at least in vitro or in an animal model system? And what we found was that in the PGC1 overexpressing muscle cells, which in many ways phenotype exercise, okay, in a dish, admittedly, that we found this small molecule called beta amino isobutyric acid that came to our attention mostly because it was the most inducible small molecule expressed or secreted by these cells, but also because we saw it was highly upregulated in acute and chronic exercise in mice. We went on to show that this small molecule, actually, if you sprinkle it on fat cells, makes them take on a brown phenotype. And I showed you that UCP1, this important factor that dissipates energy as heat, went up as much as 15 or 20-fold in mice that were given extra beta. Okay, and then finally, we went on to show that in the top left panel, that beta increases oxygen consumption. In the top right panel, that it reduces body fat, and then finally that it improves glucose homeostasis. The bottom right panel is a IPGTT, an infusion of glucose in the peritoneum. You can also do it orally, but in mice, it's easier if you give it in their bellies. And you could see that you get this big excursion in normal mice. In mice that have been given beta, those are normal mice that actually been fed a high-fat diet. If you give it if you give them beta, that glucose control is improved. Now, admittedly, this is mice. It started off in a cell culture system, and that's kind of where we were a number of years ago. It turns out that we made this observation. A French group had actually begun to study beta as well and had filed IP around this. And now if you go to GNC, you can actually get beta in a bottle. It's got a great name. It's called Lipomorph. And because they had all the IP around this, I'm actually in front of you today to talk to you about our research, because we didn't have any of that. So anyway, so beta is now one of a number of autocrine and paracrine factors that have been described in the context of exercise, either in a dish or in animals, but we got to think about humans, right? I mean, exercise is a very different beast in humans. And with that in the way of background, we really started to collaborate in quite a strong way with Claude Bouchard, who's really one of the fathers of exercise physiology, and work with him in samples in the Heritage study about 6 or 7 years ago. And we were finally funded a couple of years ago and really have begun these analyses in earnest. And that's most of the data that I'm going to show you today, as well as an update on the Motorpack study as well. So this is a 20-week exercise training study in stone cold, shall we say, normal individuals that were meticulously phenotyped after these 20 weeks. And they had submaximal oxygen uptake. They also VO2 max, they had blood pressures very meticulously taken, and many other traits, including glucose homeostasis, et cetera. And I'll walk through some of the data right now. But just to give you a feeling of what these individuals look like, there were about 745 who started. There were a little over 650 who finished. These are 34-year-olds, so these are relevant to our fellows that are visiting, or prospective fellows, and our fellows a little bit older, but still relevant, maybe less relevant to some of us in the audience. There was actually parents and children enrolled. So, so in terms of age, 34, 55% of them approximately were female. It was a reasonably heterogeneous group. BMI was 25, blood pressure was 120/60, essentially resting heart rate was in the 60s. So normal individuals, what happens after this training? So the first phenotype that we looked at was the proteomic profiles of maximal oxygen uptake, or VO2 max, because it's such a wonderful integrator. This is a classic slide from I think it's the UCSD group. I think it's Wasserman's group. Because VO2 max integrates, you have the circulation in the middle, but you have ventilation, you have mitochondrial function. And this is one of the first metrics that we look at. There are lots of metrics, and I'll tell you about some of the fellows that are working on specific projects in a little bit. But this is the VO2 max work. And this is what's called the Volcano Plot, and there's no reason to look at individual proteins. But what I'm trying to show you here is on the x-axis are relative changes, and on the y-axis is significance, and these are false discovery rates. These are not raw p-values. So lots of things are associated with VO2 max at baseline. And lots of things, a few less, but lots of things are associated with your delta VO2 max. That is the change that's going to occur. So this is a baseline sample predicting what's going to happen in the future, if you will. And all of these analyses are adjusted for age, sex, BMI, race, baseline VO2 max, and this is for the VO2 max changes, and lots of other things that the reviewers insisted upon. Okay, so really lots of novel stuff that's associated with VO2 max here. I'm just going to give you a little flavor for the biology. We were surprised by how many secreted proteins we identified that are relevant to bone homeostasis. There's a protein known as SMOC1 that's very important in osteoblast genesis, if you will. There's multiple proteins that we identified in osteocyte biology, including various BMP members and a protein known as noggin. They all fit into the same pathway. So what's really fun is that we weren't just seeing scatter shots of findings, but things coalesce into a lot of biologically meaningful pathways that might ultimately be leveraged for therapeutic interventions. Lots of interesting proteins, 4 or 5 of them associated with cartilage biology as well. Okay, so that's one set of pathways that was clearly highlighted by this work. Another interesting finding, and is that there was a minimal overlap between the proteins that were associated with VO2 max and the delta VO2 max. Remember that figure that I showed you, this classic image that everyone in exercise physiology shows almost as part of their talks, that baseline VO2 max doesn't predict the delta. And this is biochemical evidence that really Jeremy loves to show, that demonstrates that there really is very little overlap, that we're kind of talking about two sets of potentially biological predictors and potential pathways as well. So how about, whether or not we can predict those who have a particularly good exercise response? And in this case, we're looking at individuals whose response was better than a MET. Say that's a clinically relevant, a delta, I think, over the course of a 20-week training period. And, I think it was like a 15% change or better. And so these are ROC curves for changes of relative VO2 max with exercise training. So on the left is a cutoff that's using all the clinical factors in the heritage study. So if you throw in hundreds of factors, and all of this work was validated, by the way, in a second cohort. I'm showing you the work in because we had more data, and it was a more rigorous study, the first one that we did in heritage. But you could see that the AUC curve is a little bit better than the diagonal line, which is 0.5. By contrast, if you look at the clinical trait plus the proteins, there's I think a very impressive predictor that we've generated using some of the top proteins that we included in our model. And again, this was validated in a second cohort. Now, diving into some of the proteins specifically now, there's a very interesting protein known as fibroblast activation protein that we're interested in, that's positively associated with VO2 max and regulated by aerobic exercise. FAP or FAP is inversely associated with body fat. It's associated with baseline VO2 max. Even after adjusting for fat-free mass, it increases very robustly with exercise. Its increases are positively associated with VO2 max gains. And we've looked now with our colleague Paul and in other data sets, and it's induced after exercise in animal models, and then in a little bit of human data that's available. And it turns out it's actually activatable by PGC1 Alpha as well in model systems. Okay, so why is this protein kind of interesting? Well, it's a circulating enzyme, so we're very interested in these circulating factors that might be upregulated by exercise. As I've told you, we find it in the blood. What could it be doing? It's related to the DPP4 pathway, which is important in glucose homeostasis. It modulates some important hormones that are well known in diabetes. And importantly, Ellen Puré and other groups, Ellen is at the University of Pennsylvania, with whom we're collaborating, and she sent us the mice. And there are several studies out there that have looked at this in the context of adiposity and even in atherogenesis, but no one's looked at it in the context of how this might be working in terms of exercise physiology and as a hormone, and we're working on that right now. And clearly we were interested in this because it's a potential target for the treatment of type 2 diabetes, obesity, et cetera, et cetera. I'll end this little segment of the talk by looking at how we leverage other cohorts. In this case, we're looking at this specific protein, and this is how we begin to build a case for diving in deep. And we'll talk about this at some length in a little bit, but this fibroblast activation protein is inversely associated. As I told you, it's positively associated with VO2 max. It goes up after exercise. It turns out it's strikingly associated with cardiovascular and all-cause mortality in two cohorts, including the Framingham Heart Study, and this is the Malmo Diet and Cancer Study. And you could see that this is a stepwise selection, and we've integrated age, sex, BMI, smoking, systolic blood pressure, total cholesterol. We've really kind of thrown everything at this: renal function, diabetes, insulin resistance, et cetera, and it seems to potentially add information. You know, I'm not trying to tell you it's this new, we're not trying to push the marker aspect of this, but the fact that this is how we're building the case to build the biology around this, that to really inform us for a deeper dive. And for those who are interested in this study, it was published recently in Nature Metabolism. Jeremy drove these analyses and is doing a lot of the follow-up work. And we got a good shout out from Gretchen Reynolds, who's the, Jeremy got quite a nice shout out from Gretchen Reynolds, who's a writer for the New York Times. He's actually followed several of our papers, fortunately, and it's the title of the talk was "The Best Type of Exercise a Blood Test May Hold Some Clues for Us in the Future." Now, clearly, there are lots of limitations with this work and with what's going on in the field. There's very few well-controlled randomized clinical trials. There are a limited number of patients. There are a limited number of clinical measurements that have been done, limited number of molecular measurements, tissues that have been analyzed, distinct regimens. And for that reason, I'm going to spend most of the rest of the talk on the Motorpack consortium, the molecular transducers of physical activity, which is a pan-NIH study. It's sponsored by the NIH Common Fund. And as I mentioned, we're one of the biochemical profiling course for this study. So this extends work that's out there for sure. It's really a huge study. It's really up now going to be about 2,700 adults, but 3,000 total individuals. There's a pediatric arm to this as well. It's a randomized controlled study of endurance exercise training versus resistance training. I mentioned the numbers. It's 12 weeks of intervention. Again, it's completely normal individuals. Which is something we'll get at in a second. And in general, though, it is a single dose endurance, or for the aerobic training, it's a single dose. It's a supervised thing. There has to be intensity compliance. There has to be target for adherence, et cetera, et cetera. There's sort of a warm-up period, but by week three, I meant to say a warm-up over time, over chronic exercise, and then you reach, this is an example of the endurance exercise description, by several weeks into it, you can see it's about a 45 to 55 minute regimen. It's a moderate regimen. There's a cool down period. And for those who are interested, there's plenty on the website, but it's a moderate intervention in normal individuals. There's also, importantly, a resistance intervention protocol as well. We're really learning a lot about the salutary effects of resistance interventions in terms of fat metabolism and interorgan communication. And this slide just kind of highlights that this is also supervised. It lasts, it's 4 days a week, 45 minutes. It works your hips, your quads, your hamstrings, your arms, et cetera, et cetera. Okay, now what really sets Motorpack apart from any prior effort is the incredibly deep molecular phenotyping that's going on. All of these individuals are getting whole genome sequencing. It's the largest study. They're getting proteomics, transcriptomics, really RNA-seq, DNA-seq, if you will, proteomics. There's secretome studies. There's in the muscle. There's studies of the muscle and the fat. There's phosphoproteomics, total proteomics, blood proteomics, looking at all kinds of important post-translational modifications. There's studies of the lipids that are deep. There's studies of other types of small molecules. There are about 5 different metabolomics platforms that are being applied to this. And then importantly, there's a study in rats. I don't know why I used the word parallel rat study. It's as good a study as could mimic what's going on in the humans in rats. There's an acute intervention and a chronic intervention. Obviously, this is important because while this study will get lots of fat biopsies, lots of muscle biopsies, there are other organs like liver, heart, etc., that can only be done in model systems. But I think it'll help our group in particular as we try to dissect interorgan communication. And this is just, you know, in one of the earlier slides, you know, showing that the subject arrives, there's a rest period, they get a muscle and an adipose study, a biopsy, then they get a biopsy at the end of acute exercise. This middle biopsy after 4 hours has been stopped due to biopsy fatigue, shall we say, but there's a biopsy the next day, and there's actually two. Remember, you get this before and after in this case, the 12-week intervention, and lots of blood studies as well. Now, the study had really gotten a lot of steam, but you can imagine with COVID, there's been a real, you know, problem throughout the country in terms of enrollment. It had picked up again, it slowed down, but hopefully it's going to restart, you know, God willing, over the ensuing period to help, shall we say, induce that. I've recommended some volunteers. I've updated this old joke of mine, but these are our first, second year, and third year fellows. All of you have now been, your names, social security numbers, and bank accounts have been sent to the Motorpack study. I'm joking. We do have lots of, I don't have all the third year fellows, but it does seem like we do have one or two that we could lose, although some are going to be chief residents, so maybe not. So, but hope you'll enthusiastically sign up for at your nearest place. I'm going to show you one kind of fun slide. This is from the first about 900 individuals that were enrolled, and or 900 samples from individuals that were enrolled. This is just another volcano plot, again, looking at, in this case, on the x-axis, beta estimates, on the y-axis as minus log changes in blue is anything that reaches nominal significance. But you begin to put together these profiles of highly active versus sedentary individuals, and then we're going to have it before and after all these two major interventions, but we'll also have it in children as well. Now, there are lots of holes there. I mean, there's lots of opportunities. You know, these are all normal individuals. How about people that fit the pre-diabetes, you know, the DPP-like signature, if you will? How about individuals with heart failure and preserved ejection fraction? How about arrhythmia? How about peripheral vascular disease? How does that work? I mean, we know it works, but how do those individuals differ from the normal individuals that they're going to be studied pre and post COVID? We have a study with Bill Krause, who's down at Duke, and related to that, you can imagine distinct interventions. Our institution has lots of expertise in deep cardiovascular phenotyping by imaging, and I would encourage everyone to think about ancillary studies that can be used, and then using Motorpack as the comparator arm. And we have one of those studies here at our institution. So, you know, I showed you these volcano plots. You know, you can't even see what are these proteins. I've highlighted a couple of them, but, you know, you get these long lists. How are you going to begin to triage this into kind of useful information? Well, I showed you, I think, clinical associations. Hey, this fat protein, it's associated with MVO2. It goes up after exercise. I showed you its associations or inverse associations with visceral fat. And then it's, you know, we can look at it in the context of our large population-based studies. That's a great place to start, I think. But you know, we want to go back to animal studies to test for causal relationships. Some of that information is out there. Some of that information is not. And I can tell you we have 5 ongoing studies now in animal systems, and 3 of which I'm going to describe that are getting pretty far along right now. And then finally, I'm going to tell you how we use human genetics to help us triage. So this seems like an aside. I remember showing this slide the first time I ever gave grand rounds. And then I remember after me, James DeLimos came and he showed the same slide. Mike Gibson was showing that slide soon thereafter, and we've seen it a number of different times. And this is a slide that talks about what's known as Mendelian randomization experiments or triangulation experiments. Let me walk you through this and how we're using this as a framework, how human genetics can give us, if you will, a randomized clinical trial. But in this case, the randomization is what the genetics that our parents gave us. So in 2004, a group in France identified variants that were associated with premature coronary disease. And it turned out that it was in a gene known as PCSK9. PCSK9 biology was then worked out. It was clearly associated with blood LDL levels. We know the wealth of data that associates blood with blood LDL and myocardial infarction. And then Helen Hobbs found variants that were associated with loss of function and then myocardial infarction. So clearly it became clear that in independent analyses that PCSK9 is a driver in this triangulation experiment of myocardial infarction itself. So now that's using a drug that was described using human genetics and intermediate phenotypes. But in our case, we're looking at human genetics, relating them on the bottom left to intermediate phenotypes. They can be chemical compounds, they can be proteins, they can be lots of other intermediate traits that we have that are unique in our cohort. And then we ask, are those same genetic variants associated with diabetes, atherosclerosis, MVO2, brown fat? So I showed you, hey, these associations, brown fat, and its associations with outcomes, it's kind of interesting, lots of confounders. But what if I were to tell you that a master regulator, a gene that's important in brown fat and how it's generated in human development is also important in cardiometabolic disease? Well, those data are coming, and I would tell you to keep your eyes peeled. It's not from our group, but I can tell you that those sorts of analyses, these sorts of triangulation experiments are what we're trying to do. And we have lots of examples now. Yesterday I was talking about this with Mike Mee in the group, and Mike said, I think we only have one where we have the triangle here. And I said, Mike, we have one with the triangle. That's fantastic. We have identified a new factor associated with glucose homeostasis and diabetes. That's enough to go to town on this factor. And what we're doing is we're leveraging lots of data that we've generated in the Jackson Heart Study, in Mason, the Cardiovascular Health Study in Framingham, and that we're working on in the cloud at our institution and at the Broad as well. So let me just tell you about some of the ongoing studies. I'll end in the last couple minutes. I've lumped individuals into these sorts of questions, but lots of these go beyond the individual questions that for which their name is underneath. But do these markers predict outcomes, or can they predict, more importantly, the response to therapy in heterogeneous patient cohorts? So Dan Cruz in the group is doing some really interesting work comparing and contrasting genetic findings and social determinants of health and that interaction, which is obviously a critically important area. Zuzu Chen, who's an endocrinologist, is looking at outcomes in diabetes prevention programs. She's had a couple papers in the DPP, has another paper that's coming out, hopefully, so that we're working on now soon, at looking at diabetes traits with a particular focus on interaction of therapy. Sure, it's nice to have a predictor. She had one paper where she described some lipids that baseline levels predict your response to the lifestyle regimen. And that was, again, published in Diabetes. Mike Mee is very interested in diet in general. I stuck glucose trace, and that's what he's studying right now in Heritage, but Mike is our resident expert in all things related to crazy diets. So if you have any questions, ask him about those. I think he just quit his intermittent fasting diet, but he can chime in if that's otherwise. Prashant Rao is studying cardiovascular responses in heritage. And then there are lots of questions that I've alluded to around the genetic underpinning of these factors discovered in these studies, but also in other studies like Jackson, where we're beginning to look at where we're looking deeply at human genetics, and does modification of these pathways modulate exercise or cardiometabolic traits in model systems. Mark Benson is interested in a circulating enzyme that's important in branch chain amino acid metabolism and overall amino acid metabolism. We have those mice, we're working on them. Usman, who directs our cardiovascular genetics program, is looking at a really interesting novel predictor of cardiovascular disease that we found now in 2 cohorts. The mice are on their way, and Usman is really going to dive into some of these studies in collaboration with Dongxiao Shen, who's been leading a lot of the animal work in the lab for a long time. Dan Katz is looking at genetic architecture, working with the Broad. He's now doing his heart failure fellowship, but is deeply involved in the group. And I've highlighted Jeremy's studies throughout. I also want to thank Lori Farrell in our lab, who is really underpins so much of the work, not just the clinical work, but a lot of the profiling and to help really bring this all to fruition. And I want to thank her in particular as well. And then finally, a welcome to the fellows who are here. And I want to put in a word. I don't know if Jackie is on the line, but Jackie White, who runs our fellowship program, she's probably not going to hear these comments, but she has organized the day today, and the passion that she puts into the fellowship is second to none. I'm going to brag and say that she's the best in the world. She is just so special, and we're deeply appreciative of her efforts today, and really underpinning so much of the interface of what we do in the training aspects of our program. So thank you very much. And maybe I'll answer one question. We do have interviews. So maybe one, if anyone wants to ask a softball question, I'm just kidding, please do.
Thank you very much.
May I ask a question?
Absolutely. Go ahead, Dr. Savitz.
So Rob, that was wonderful, of course, and it's so great to have the Chief of Cardiology kick off the Grand Rounds series with a spectacular lecture. So, I study arrhythmogenic cardiomyopathy, which is one condition in which exercise seems to be a bad actor, in which it clearly increases risk of adverse events and increases penetrance of clinical expression of variants. So I'm just wondering if in the Motorpack study, it might be possible to look at individuals who may have variants in desmosomal genes who may not have clinical expression of disease, but to see if they have a different response to exercise. And a related thing is this, there's a paper that Matthias Narendorf published a couple of years ago in Nature Medicine showing that exercise changes the inflammatory cells in the heart and mobilizes cells from the bone marrow that help the heart experience beneficial effects. So I'm just curious if it might be possible to do that as a sub-study in Motorpack.
So right now, Jeff, I can tell you, to the best of my knowledge, there are 0 studies. Let me repeat, 0 studies on arrhythmogenesis in that group. Now, remember, though, they're stone cold normal individuals, and we should think about power calculations of, but we will see novel variants in these individuals. And broadly, will we see proteins or metabolites that are associated with these individuals? Or more broadly, Jeff, will we find acute changes in proteins or metabolites that we then test on their function? We've started some of these studies with Arti. Dan did a little bit of this that might be modulating, in zebrafish heart rate, but what's the model? Do we patch clamp cells, et cetera, et cetera? Where, Jeff, we called you about one variant and its association with proteins. You remember this a long time ago, and we're kind of getting back to this in a in a similar sort of situation. And we have to think about numbers and powers, et cetera. But this is completely ripe. And I know that I'm going to get some emails from Al and Peter afterwards. I've told you a million times, why aren't you studying arrhythmias, et cetera? It's really hard. I'll admit that one of the reasons that it's hard, the ends are a little bit difficult, right? And the studies are messier in terms of getting baseline samples, but this does provide us with a large population. Jeff, I would bring this out broader and say, is this analysis that we should be doing throughout all of TopMed, right? Where you have whole genome sequencing now in 150,000 individuals. And then we're going to have metabolite data in 20,000 individuals by the end of the year, protein data in, I forgot how many thousands of individuals, et cetera, et cetera. So the answer is absolutely. And there's no one, let me repeat, no one to the best of my knowledge looking at arrhythmias in this area.
Great, thank you.
All right, so if there are no further questions, we'll just wrap it up a little bit early so that our candidates
I've actually got a contentious question as usual, Rob.
Go ahead, Dwayne.
For procedural types of things, we always look at net clinical benefit. Everyone's hassling us about indication. Should we be doing things? Of course, I can get the blood vessel open. Of course, we can get people to exercise, but I see you hobbling around after you go for a run. I see my friend walking around in his boot after his Achilles tendon rupture. Jeff talked about the person who drops dead on the soccer field. I appreciate the deep dive and the mechanistic approaches, but are we going to take a Smith Center approach to exercise of, does it really work for the uninitiated, the person who gets a new Peloton instructor and has a near-death experience, the person who really shouldn't be exercising, but is told because everybody says we should exercise?
Right.
When are we going to get to a point of, are we testing that the intervention really works?
Yeah, no, I think it's a great point. I mean, and I think that even with the numbers that we have, 3,000 individuals in a study like this, it's going to be hard to look at those sorts of, outcomes. But, it's a great question. Now, we're moving in other areas and integrating some of our genomics work and trying to do that as much as possible with some of the outcomes that are being adjudicated at the Smith Center. Jordy's working on a project. We've got a project around COVID with Kazi and Rishi and Bobby. It's a great question. I think, though, Dwayne, those kinds of questions are only going to be accessible when we have larger numbers. There's plenty of outcomes, but I think that those outcomes and the data sets are available. We're going to have confounders that those guys know a lot more than me, but I think you're going to have to then have overlay on that some genetic information as well in these cohorts. It's coming, Dwayne, and maybe those are the types of studies that can be done by 23andMe right now, but ultimately by the All of Us study at the NIH and other TopMed, et cetera, et cetera. So it's a great question. How to individualize, personalize. We need way, way, way bigger numbers. But Dwayne, until right now, the largest exercise intervention study that was meticulously phenotyped was 700 individuals. So great question. All righty. Thank you, guys.
Thank you, Rob.
I'm going to have a beer tonight instead of exercising until we get better data.
All right. Bye, everyone. We'll see you next week.
Thanks, Rob.
Bye, guys.